Good afternoon, everyone, and thank you for joining in. It's my absolute pleasure to introduce our speaker for the day, Dr. Adam. He's joining us from University of Southern California, where he's an assistant professor in the Department of Quantitative and Computational Biology. Adam did his PhD from Imperial College London with Dr. MacLean in theoretical systems biology before moving on to do postdocs at University of Oxford and then finally at UC Irvine with Dr. Jim Nine. I met Adam when he was just starting his position and had organized a single cell workshop. And I was a postdoc and I was trying to get wisdom out of him in what it means to start a lab as a junior PI. So it's great to see that Adam since then has developed a very interesting research program at USC, focusing on mathematical and computational modeling of data sets, including single cell, trying to understand cell fate, transitions, signaling dynamics, multi-tissue dynamics, and applying it to a range of different applications from aging to cancer. So with that, I guess we are very excited to have him here and listen to his talk on GR and inference. Please join me in welcoming him.
Awesome. Thank you so much for the invitation, for the introduction. I'm very excited to be here and talk to you today about some recent work from my lab at USC on inferring gene regulatory network dynamics that control cell fate decisions in single cells. So very broadly, my lab focuses on understanding cell fate decision making. So just to get us all on the same page, I would like to think about a survey of possible cell fates. That is, what are decisions that a cell can make? So this is my cell. And we're going to ask, what can it do? The first thing is it can do nothing. This is very important. It can be an active choice and an essential one. The next decision is of course, it can divide. This is essential to all of life. My cell can die. This can also be a very active choice by many different mechanisms, and it's essential to multicellular life. And then my cell can do things like migrate or transform from one state into another. And finally, it can differentiate. And I separate this fate from the others because it can, in a sense, be made up of these more kind of fundamental cell fate decisions, but I include it because it's so essential to all of stem cell differentiation, which is a big topic of the research in my lab. So this idea of cell fate decision-making really came from the inceptions of the field of stem cell biology. And stem cell biology really began, well, roughly exactly 60 years ago. There were a few papers in the 1960s by two Canadian scientists, Till and McCulloch, in which they both through a mathematical model and also by isolating cells from the bone marrow and culturing them in a dish, they found these cells which had this ability of high proliferative capacity, self-renewal, as well as being able to differentiate into specialized lineages. So I'd like to, I think it serves as a nice reminder that the field of stem cell biology was theoretical and mathematical from its very earliest inceptions, and it was actually years, 25 years more before stem cells in any organ were actually isolated and proven to exist in vivo. So the stem cells that I work on primarily are blood or hematopoietic stem cells that reside in the bone marrow of adult mammals. And you can think of the production of the whole blood system as this hierarchical tree, where you have an HSC, a hematopoietic stem cell at the top of this tree, and through successive divisions and differentiations, bifurcations, through all these blue populations, which are progenitors, then they form all the differentiated cells, the erythrocytes and platelets, and the myeloid and lymphoid cells of the immune system. In the last 10 years or so, since single-cell genomics has been around in high throughput, we've learned a lot. And we've learned that, in particular, there's much greater cell heterogeneity and much greater cell type plasticity than previously known, meaning these transitions do not always go one way. They may be reversible, there may be trans differentiation, you may be able to go sideways along this differentiation hierarchy. So I'm thinking in terms of, for example, trees, which are like necessarily non-cyclical, it might be problematic for this biological system. And I'm going to talk about aging. So as we age, stem cell functions change, including these HSCs in the bone marrow. Actually, a lot of things happen as we age. A few of the things that happen include you have this skewing of differentiation. Rather than having the balance that your blood system wants for a good function, you start getting too many myeloid cells and too few lymphocytes. You also have a growth in the number of stem cells, but reduced polyclonality. So these start to evolve somatically inside the bone marrow. You also have changes in metabolism and a lot more, some of which we'll get into today. So we really started off with this question of what minimal gene regulatory networks control stem cell fates in this differentiation hierarchy, and how do these change as we age? So to study that, we began with the knowledge that changes in early life can affect stem cell fates as we age, particularly perturbing metabolism or diet in early age can change the function of stem cells as we get old. But we don't know what networks are required to control these dynamic stem cell fate decisions. So to study this, my collaborators at the Fritz Lippmann Institute in Germany for stem cell aging, they have this experimental setup where there's 4 samples. We're going to look at stem cells sampled from young, like roughly 6-month-old, and old, like 2-year-old mice. And these mice are going to be either fed anything they want, or this DR treatment, dietary restriction, is basically 70% of all the calories they would want. So it's a mild dietary restriction for 2 weeks, which we've seen in previous work induces a functional change in the stem cells as the mice age. And we're going to study this with the 10x multiome platform, which assays both the single-cell RNA-seq and the ATAC-seq to quantify chromatin accessibility in the same single cells, and then perform network inference on these joint data. So just to go a little bit more into the data, these joint multiomic data, they offer a new resolution with which we can look at cell states. And this is actually not from the current data set. I'm presenting to you as an example just to get a feel for these data. So single cell gene expression, which you're maybe more familiar with, is a noisy measurement, but one which allows you to measure transcriptional state of every single cell for hundreds of thousands of cells. Now what we're doing is we're measuring along genomic intervals, are there peaks in the chromatin, which gives us an idea of the accessibility. So the types of things that we can do this for, this data set comes from the mouse kidney. And this, like with the universe approach, is like data integration, and what it lets you do is it lets you refine the cell types with better accuracy than using just the gene expression alone. And then over here, so I'm showing you just the track for the chromatin accessibility in one cell type, so averaged over single cells for one gene, this ABCC3 gene. And now this is the female samples, and you see a strong peak here. This is a female-specific gene, and basically in the male samples, there's no peak in this promoter of this gene. And then what's really interesting in the study, we were looking at the sex regulation, and basically by knocking out androgen receptor, which binds testosterone, then you can see this partial recovery. So we're understanding through this that the effects of sex dimorphism in the mouse kidney is in part regulated through this expression of androgen receptor. Right? These are showing the gene expression distribution. So by using this multiomic joint readout, we're really able to dig deep into the gene regulatory mechanisms, in this case, controlling the sex dimorphic function of the kidney. Okay, so we'd like to perform GRN inference. And it's a very reasonable question to ask. Why develop a method? There are a lot of methods out there. Where does our multiomic approach fit into the current state of the art? Well, one important factor. So we would like to use, we're very interested in how cells change state over time. a very good way to get at that is through trajectory inference, or the use of pseudotime. But many other works have shown that it's really hard to do that well. In fact, sometimes using the pseudotime makes things worse than random in a way. And yet we still really want to use it to account for these stem cell differentiation dynamics. Another important part of this is that we want not only directed networks, but also networks that are signed. So TELS activating or inhibitory relationships, which can be quite hard even for some of the kind of newer methods that are using data in different ways. And we're also looking for interactions in hematopoietic stem cells. This is a very specific cell state. And so if we have this strong prior on like transcription factor motifs, then we're likely to get the types of things that are enriched for the types of transcription factor that everyone studies. And that might not be what we're looking for. We're looking for something new. So we don't want to put too strong a prior on the types of gene-gene relationship. So this is our proposed method. It's called POP-infer, gene regulatory network inference with pseudocells over pseudotimes. That makes the POP. So I'll explain the two big words in that sentence slowly now. So the first is the pseudocells, which is other people call these meta cells. The idea is with the gene expression in single cells, let alone with the chromatin accessibility, the data is very sparse. And without some sense of pseudo bulking, you don't have enough signal really to work with there. So it's just very simple here. We bin cells, K cells into like equal bins over the pseudotemporal trajectory. And so then everything I show you in terms of the plots, it will look like single cells, but it's actually these pseudo cells of approximately 50 cells. Now, what are we're binning over? That is the pseudotime. And the idea here is that we're really interested in these dynamic relationships between transcription factors and the genes that they regulate. And we've got this joint multiomic measurement. So the PhD student, Megan, who came up with this method, she proposed that the time delay between transcription factor and target is shorter when you're looking at the accessibility of these regions the transcription factor has to bind than the gene expression of the target, and trying to kind of take away that time delay and look at the RNA of the transcription factor and the accessibility of the target are these more contemporaneous measurements. That's the key assumption that we put into the model to use the gene expression of the regulator to try to predict the accessibility of the target. So here I'm just giving you an example of the data. This is just like some genes. Then we're going to have, now I'm plotting this among single cells and the pseudocells over pseudotype. This would be the gene expression, and this would be its accessibility. You can see there's clearly a correlation, although there's other steps to their relationship. And then we're going to learn a regularized regression model, a lasso model, where the one kind of non-standard part in this is gene-specific sparsity, so the lambda here. Otherwise, this is really like a standard lasso, but the lambda we're going to vary, and the reason for that is illustrated here, right? For a given fixed lambda, I'm showing you 4 transcription factors and the number of targets they get. Here, like this is okay. I mean, it's a lot. Probably these are like a lot of these should be 0. And then we get for other ones, you have these super sparse. Like the first one is actually the intercept. So this is 1 and this is nothing. We don't want nothing per transcription factor. We'd like to infer more relationships than that. So we're just going to do this sub-optimization to look, instead of lambda, we're going to replace it with an alpha, which basically just balances the error and the number of non-zero coefficients. And then we're going to look at the consensus. So some networks, for different alpha, sum the networks, varying the sparsity, and then take the consensus network W as our final network. And we found in doing that, oh yeah, this is just showing you how it looks. Basically, it's nice that you really do, as you do the lambda, you do get like a minimal that balances this optimal error. And then by this choice of consensus, sparsity, we get something that looks much more reasonable in the sense that we have a few targets for each transcription factor, which are non-zero interactions. So coming back to hematopoiesis. This is just an overview of the data we have in these 4 samples. So young and old, fed whatever you want versus diet restricted, ad libitum versus diet restricted. So we look at the multiomics data, we cluster these cells. Like these are very subtly different cell states. So we have to be very careful with how we cluster. We have to regress out several kind of confounding variables. And in doing so, we get these like HSCs, yellow, and multipotent progenitors, etc. And here I'm just showing you a selection of genes that are differentially expressed between the HSC stem cell population and the early multipotent progenitor. We can see that this expression signature is maintained throughout diet and age, although as you'll see, it is subtle, right? This is not like comparing brain and bone cells, but these are subtle cell states that we wish to look at the regulation of. So first, a couple of slides on some attempts at benchmarking with hematopoietic data, the ability for pop-infer to infer networks in these early cell fate decisions. So here we're looking at the transition from stem cell to multipotent progenitor. And here we're comparing via methods, pop-infer against these other methods which do not use the multiomic assay. So they're RNA only here in inference. And the area under the early precision recall curve is using a chip data set on all hematopoietic cells. So we're looking at a very small subset of that. So like our absolute values are very small, but relatively speaking, we're curious how we're doing in terms of the number of correct interactions inferred. And the top row is for the stem cell transition, the bottom row is for a different lineage. This is HSC to the granulocyte monocyte progenitors, or GMPs. Again, using the RNA only methods. We also checked what happens if we like scramble the pseudotype. This is the non-scrambled, and then down here. So we're doing better than if we scramble pseudotype. And you might say, well, duh, like if you weren't, that would be pretty bad. But why did we do this? We did it because previous work showed exactly that. And basically, here's the average early precision values for 3 methods for the real pseudotime, which these methods are using. And then here they do, okay, this is still maintaining the order. This is not. This is like scramble. And it's not worse for any of these 3 methods, right? So in this case, the pseudotime is really not giving you synchronal. It's basically just giving you noise. It's not helping. And since we want to use it, so we know this is a risk. So we want to make sure that there is some signal in the pseudotime, which there seems to be. So here is another comparison. Now we're comparing methods that are using the multiomic, the chromatin accessibility jointly with RNA, as we are doing, although they're all doing it from very different ways. And since there's very different strategies, it's extremely hard, like even with a chip-seq ground truth, which in itself is, I think, quite shaky grounds, it's very hard to basically like normalize these and calculate kind of AU-PR type curves. So we're doing a different approach, which is looking at the setting a strict threshold for the number of interactions, and then just asking what are they for the top transcription factors for pop-infer, and versus 5 other methods, all which use information from the multiomic readout. And one thing you'll see, so red is activating and blue is inhibitory. So pop-infer, like dictus, but kind of unlike any others, it's quite balanced in terms of activating and inhibitory. So you are getting both. Some of these methods basically like don't infer any inhibitory edges. But the more important thing is the specific transcription factors in these networks. So the majority of these are hematopoietic specific. And that's just not the case for, with the exception of big R is also capturing hematopoietic transcription factors. But elsewhere, when you see like RUNX, ETS, zinc fingers, these are things which are very non-specific. And that's not surprising because these methods built a base GRN and then did some regression on top of that. And we think that's biasing you away from cell type specificity, which is exactly what we want. So, oh yeah, finally, before we get into the kind of biology that I'd like to talk about, one more comparison between the methods. There's a data set, I forgot to put the reference on this slide, but we took validated targets, a small number of validated targets of MECOM from previous paper. And here we're just looking at the percent of correct targets inferred by top infer versus all methods. So these are RNA only, and then from cell oracle, these are using the multicellular. And so both 1% correct, and also like FigR is doing quite well, but FigR just infers a huge number of interactions compared to other methods. So it gets a pretty high false positive rate, and we stay very low. So we're kind of building confidence that perhaps we're looking at cell state-specific networks of interest. And now we're going to zoom in on our specific question, which is what is controlling the transition from stem to multipotent state in this lineage? So when we look at just some network statistics, 2 genes jumped out by their kind of ranks, both network hubs, and those were CDK6 and MECOM. And now I'm just showing you the networks, basically like intersections of young networks with and without diet effects, or old. And basically, all these interactions are actually inhibitory in this case between the 2 hub genes, and GXNAP was added because it was the only other direct regulator of these hub genes. And we see this consensus network, which is represented at the bottom, which consists of a mutual inhibitory loop between CDK6 and MECOM. And there's also an inhibition on that from TXNIP. And we've added this. These are the res interactions are predicted by top infer. We've added this black dashed arrow, which is of great interest to us because IGF signaling is like the main metabolic factor, which is previously shown to be very important in maintaining kind of HSC or HSC phenotypes that are in young life that are lost with age. So, and as you can see, like this network, this is the network that is maintained, the consensus network in young age, and it is also, these interactions are lost with age. So our hypothesis, our theory, based on these predictions, is that during the HSC to multipotent transition, when you have this MECOM high, this is the stemness factor, the HSCs are into or are kept in quiescence, and they're not able to differentiate. And if that is repressed by CDK6 and CDK6 turns on, then HSCs are able to exit quiescence and differentiate in young age. And this is broken by some extent when the mice get old. And just showing you some of the data of underlying these crucial genes, you can kind of get a taste for that. So like this is, the top row is young, the bottom row is old. So you like, this is the TXNIP, which is inhibit CDK6, accessibility, and then the gene expression of CDK6, which is to inhibit the MECOM, this is the accessibility. Okay, these are the relationships that are maintained, and you can kind of see actually how they're broken. So there's no more a relationship here, inhibitory interaction, and there's more cells which are turning on CDK6 early in age. And that's exactly what we would perhaps expect from prior knowledge, which is that these stem cells are less able to stay quiescent and maintain their states for a longer time. They're actually more active, more cycling, and they're potentially more susceptible to exhaustion earlier. So basically upregulating cell cycle related factor. So we're quite excited by this small network, mutual inhibition, and it's very much an active kind of experimental goal in the lab for our collaborators to test these inhibitions of CDK6 and MECOM, which we're doing currently in vivo in mice. In the meanwhile, we have this evidence, which is still very like, correlational. It's not, yes, proof, but we just looked at a different orthogonal data set. Gene expression is what allows us to quantify the cells into a small quiescent stem cell population, the rest are non-quiescent. I'm just showing you a couple of quiescent HSC genes. And then we looked at the gene expression of our 3 genes in the data set and saw that these were differentially expressed in the direction that we would predict from the model. So the MECON and the TXNFP are both up in the quiescent population, and the CDK6 is down. So this is, again, a nice indication, certainly not proof yet, and those experiments to try to test this in vivo are ongoing. Oh, and then there's one other piece to this, which is, so while a preprint was submitted a year ago, we're currently working on the experimental validation part, and this paper just came out over the summer, which is talking about the role of CDK6 in quiescence and blocking HSCs from entering into differentiation, which is also, this is, by the way, like, this is kinase inactivated. That means this is not CDK6 doing its cell cycle role. This is an additional activity of this gene in mediating properties of HSC stemness. So this is, I think, also quite really encouraging that people are looking at this, and in the sense it kind of corroborates some of our ideas, but we go a step further in the sense that we're able to tell you what CDK6 is, through whom CDK6 is acting to mediate this kind of quiescence-related activity. Okay, so we're looking carefully at these learning-directed networks from data, but we're also really interested in kind of going a step further and asking if we can learn parameterized models. So again, just, this is data I showed you before, but let's pretend we have something new here, and we're looking at some dynamics of species, biological species. I'm actually being really agnostic now whether these are cells or genes, something else. And we're looking them over a trajectory, which could be real time, or it could also be pseudo-time. And the question is, can we learn a model? So in a sense, this is still network inference, but we're actually interested in learning a mechanistic parameterized model from these dynamical biological data. And so that's made possible by symbolic regression, amongst other methods, but Cindy for symbolic regression, which was introduced a few years ago, provides a solution to that. And this is a framework that we came up with, where we're interested in quantifying the ability which we can use symbolic regression to perform data-driven model discovery for noisy biological systems. So the idea is we're going to take some noisy data, we're going to learn a hybrid dynamical model, which just is basically we want to learn the derivatives from this data using a neural network, but we have some knowledge about the biological system. We don't want to throw that away. So the G of X is like known interaction terms. We want to put them in. Everything we don't know will be put into a neural network in order to learn the derivatives. And then based on that, we're going to do symbolic regression using Cindy to learn the mechanistic form of the model from the fitted dynamics. How we deal with data is just take multiple trajectories. These are like raw, noisy samples from a biological system with multiple species, and we're just going to kind of take it, put it into batches, and then train over these batches with a sliding window to account for the different sources of noise. And then we're just going to train quite a simple neural network model on this unknown dynamics, where we're using an ODE solve and training that by Bragg propagation. Eventually combine it with G, the known part, and then just solve that predicted F to input it into our, this is basically the Cindy problem for symbolic regression, which takes some X prime, some derivatives, and seeks to learn like the optimal coefficient matrix psi, which minimizes this, where these are the coefficients, and this theta is a basis of model terms. So basically, it can learn an interaction model. You have to input what could the terms be. So they might be linear or polynomial. They can be more complex. So you have to give it a space of possibilities. And of course, if you don't have the correct terms in the basis, then it cannot come up with the correct terms. And also, if your basis is very large, computationally, that will become hard. But in theory, this can work very well. So let's see how we do in practice. So why do we do this hybrid approach, right? Where hybrid here means we have this known, a known, and a known. So we just, for a lock of Altera predator-prey model, we test first just base Cindy. So just you put the dynamic trajectories into Cindy for symbolic regression, and it goes okay. I'm showing you both for both additive and multiplicative types of noise. These are the learned derivatives, dxt, but you can see like here at the end, like it starts to miss. It starts to infer this derivative less correctly, and that's also the case when we just try to fit with a neural network with no g of x. We fit the whole thing. Again, it does not too badly, but at some point when the derivative gets larger, it does less well relative to our hybrid model, which has actually managed to fit a really pretty accurate derivative throughout the whole time interval of this training, which is 4 seconds. So then using that hybrid piece here, we're going to try to learn these lockable term models to start with a simple example, different noise levels and different noise types. We're interested in both additive noise and this multiplicative noise, which scales with the species. So I'm showing you additive noise here at 1, 5, and 10%, and then the same for multiplicative noise at 10%. So it does really well. At 1 and 5%, it's very good. At 10%, it starts to introduce terms which are incorrect, as well as the correct terms. I'm saying correct, like when these are not like the actual value is like 0.9. So this is not like, but it had no knowledge of this interaction term. So I call this correct. It's pretty damn good. It's not 0.9. And this should be 0.8. So it's like 71 or 0.814. So these terms are still pretty close, but it's also introducing some wrong terms. At this, we introduced like a model selection scheme over the hyperparameters of the machine learning to learn the best model. And here, this is like the model with the lowest AIC. And it does have this extra term. When we look down, so basically this whole idea of Cindy is like it can propose models. And will it always get right? Like the rank number 1, the correct model? Well, no. As the noise gets high, like 10%, it will not. But when you look down the rank, it does have correct models. It's just, right, because we're training on 4 seconds. Now I'm extrapolating out to 20 seconds. So these were lower error on the first 4 seconds, and unfortunately they did not contain the correct terms. And you can see that obviously they don't oscillate with the right amplitude, or in fact damped as you go out. So these models, while ranked slightly worse, they actually did contain basically the right terms, and then they extrapolate much better. So you can't just take this off the shelf and blindly choose the best model and run with it. But it's, I think, a really powerful way to propose models, maybe that you take a small set of highly ranked models away to start to test in different means through further experiments. We tried it on a model called the repressilator, which is a synthetic oscillator from a quarter-century old paper from Michael Ellis, where it's these 3 inhibitory interactions. It can be represented by gene protein, or we're just using this like protein only, like a simplified model. So it's a 3-species model. And basically, like this is just simulated from the ODE model. 3 coupled ODEs where we have these it's like non-polynomial here. So you have these Hill inhibitory interactions in the model. So now, right, if you do this, if you have lots of Voltaire data and you plug it into symbolic regression in Cindy will do very well. It will get you the right answer, even with not too, with a little bit sparsely sampled templates. That's no longer the case here. Now we plug the correct, like the noiseless data into Cindy, and it doesn't learn the derivatives well enough to get the right model. it's got these, the purple terms are all incorrect. So it's like learning correct terms, more or less, but it's adding extra stuff. So it can't even do this on the noiseless data for a model which is this like dynamically rich and complex. So it kind of provides an additional motivation to use intermediate method to get better derivatives. So that's what we'll do. And using the hybrid dynamical systems. Now we're looking at, again, noise levels from like 0.1% up to 10%. And we find, for both additive or multiplicative noise, then we are able to learn correct models. Here we're learning this pill interaction term, and the basis consisted of, we told it that it should be a Hill type term, but we didn't know whether it would be what the order was. So the Hill coefficient could be 1, 2, or 3, and the obviously the coefficient could be any real number. And the correct coefficient is 10 for each species, and you can see it's like the error on, again, this numerical coefficient is increasing somewhat, especially here at 10%. And these are not, again, the top ranked models, but we can find correct models even at 10% noise. And the final example is on real data. So up until now, these have been synthetic data examples. We're now looking at some single-cell RNA-seq, specifically describing the epithelial to mesenchymal transition, or EMT. So we have this epithelial cells can turn into mesenchymal cells for development as well as cancer metastasis, and they can do so, and they go through an intermediate state. They can also transition directly, although in the case of our data, we see evidence for this orange intermediate state. So these are data derived from single-cell sequencing, where we're quantifying the cell states over pseudotime now into these 3 cell states: epithelial, intermediate, and mesenchymal. And we're going to feed these data into the hybrid dynamical model, also comparing that to just like directly feed this into Cindy or directly learn a neural network. So Cindy really struggles to find any model that even this is, within this is actually quite a large basis now, that it just doesn't find a good fit model that can simulate data which is consistent with these data. The pure neural network approach does pretty well. You can kind of see, I think there's like some evidence of overfitting here. There's some parts which just look a bit overfit. And then finally, this is the hybrid model simulated trajectories compared to data. So the equations here are for the fitted hybrid model. And like we also have, we had some prior knowledge. So we had our own model. We didn't tell Cindy that, but so it's interesting to compare our own simple model of the cell state transitions through this EMT with that which we can infer directly from data. We see that there are basically there are known interactions which agree, and then there's some regulations, there's some evidence basically of this direct transition which we did not necessarily expect to occur. And then there's also a nice part of this, which is that you can actually now let me see if we can see where it is. But we added this degradation term to each species because we believe that each cell state can degrade over time, can be lost over time. But our degradation term, then you have to parameterize that, and our degradation term was too high. So the Cindy actually basically compensated for that. So it balanced it out to reduce the degradation overall. So you can actually, it's almost like a different way to set a prior. You can make a guess about a model interaction term, and then as long as you put the same term in the basis, then the symbolic regression can compensate and say, if you're wrong, then I'm actually just going to add a the same term with the opposite sign, cancel yours out because you're wrong, and that shouldn't be in the model. And I'm going to tell you what should be there.
So is this application one individual or
in the app, these are from a population of cells which are stimulated by a TGF beta
from one individual,
from one cell line. Yeah, but we have data from many cell lines and many stimuli. So yeah,
so
we're working
are
on
you trying to integrate those? Or?
So yeah, exactly. So this is because the responses are heterogeneous, but certainly a goal is to learn a consensus model which is not specific to the single cell line or the single stimuli. Okay, so basically, in terms of network inference, I've talked about this method, pop infer, which can infer GRNs using multiomic RNA plus attack-seq data over dynamic cell state transitions. It does well and outperforms methods that don't make use of the multiome, use RNA-seq only, and we believe it's better in the sense it's more cell type specific on these multiome-based methods when we're looking at HSC, hematopoietic biology. We've used this in application to stem cell aging and hematopoiesis to discover this GRN, which controls stem cell quiescence versus the genes MECOM and CDK6. And I've also talked about this extension to like parameterized DiffEQ models and how these can be learned using hybrid dynamical systems from noisy biological data, including from single cell RNA-seq. And yeah, both the code is on the lab GitHub if you would like to explore those more. Now, are there any questions in the first part? Yes.
Yeah, I have a question about like how you build the up in the cells when you did a pocket for like what method did you use for aggregation? Is it like epigenetic or like is it like you take the number of counts? Like when you do aggregation, like did you type it out or like did you?
The aggregation itself. Yes. And it's so it's not an aggregation, it's a sum. We sum the counts. So because we want to yeah. So yes, it is, sorry, average. No, yes. We aggregate, we do the sum, and it's defined based on, you have to have like a similarity metric. So like how are the boundaries of pseudocells are defined by the pseudotime. And then within each pseudocell which is defined, then both the reads that map to attack and RNA are aggregated. Yeah.
Thank you.
I just missed this part. When you say you make a pseudocell and you're adding up the counts, you start with single cell data and then you make a multiomic data, or sorry, can you clarify?
Yes, the data are single cell, and the pseudocell, so it's aggregated both, so per gene or per gene accessibility over however many cells are in the aggregated pseudocell.
Okay. Oh, so you aggregate
for every single gene or accessibility region.
And I'm particularly interested in the question model. So the first question is, I see that is the gene accessibility score request on gene expression level. Have you seen other programs they do it in the inverse way, which is like using the gene accessibility to predict the gene expression level? So is there some like more biological insight about it? Which should we request on and which should we? Another minor question is, have you done any like denormalizations or descaling to divide it before you put it into the model?
So what normalization is done on X, the gene expression data?
Yeah.
I mean, we do log normalize those data. Yes, we don't scale or anything like that, but we log normalize the gene expression data. I didn't quite give you the details. The gene accessibility is its own calculation. It's actually like a per gene accessibility score. We're not looking over, again, these data are incredibly rich. We could look at so many more things, not just promoters, but enhancers. I mean, there's so much in there. For now, we're not looking at non-cis regulation. We're calculating gene accessibility scores, which is like this function which is centered around the promoter and then contains some upstream and some gene body region. So there's a one-to-one like accessibility to gene mapping. Of course, you could do more complicated things. And for the regression, I guess I think the key thing here is like the sense in which we're using the dynamical information. That our prior expectation is that, and indeed we see this in the data, accessibility can, changes can precede expression changes. It's what's like the thing that convinced me that this might work is we looked at some lineage marker genes, and the thing that's supposed to turn on, the chromatin around its promoter opens, and then the thing turns on. So I think in our case, it would not make sense to do the other way around. But for non-temporal data, then you have a different situation. Then I think then it would be reasonable to do it either way. You could predict on the inverse. Yeah. I think I don't have enough time. I thought I spent a lot of time on the first part. So I don't want to go over time. So I want to tell you a little bit about clonal hematopoiesis. And basically, I'll just tell you the punchline. Basically, there was a paper 10 years ago now that said that 2/3 of cancer risk is bad luck, can't do nothing about it, because they did total stem cell divisions by cancer type against total lifetime risk, and they got a linear correlation. Got a science paper for one linear correlation. you do if you're Bert Vogelstein. And then there's a big fight about it. But basically, the takeaway is that stem cell divisions over a human lifetime really matter. Okay, however much you think it might influence cancer risk. And so that's true in the hematopoietic system, where we see this massive difference. On the right are the old individuals. They have used large clonal expansions even when you don't get cancer. The question is, when does this variation arise? And I'm happy to chat about this more. I'm going to skip all over the details, but basically we use this fluctuating methylation clock, these neutral CpG sites, as a lineage tracker. This is an example of flip-flopping methylation by Jay, lead author's dog, who's very good at fluctuating in terms of his methylation status. And we use this to model these clonal dynamics in the stem cell niche. We model it from development all the way through life. And basically using that plus twins data, where you have this shared genetic component or shared blood, in the case of monochorionic twins, then you find that you get these different correlations in the clonal composition. So basically, this is unrelated individuals, you have a lower correlation than with twins, which have a high correlation in clonal composition. And that's like this. These are wild, because these are 90-year-old twins who have like a 95% correlation in the clonal composition of their bone marrow. Right, that's pretty wild. And so where does that variation arise? Using these twins data, coupled with this really neat clonal tracker by methylation, we're able to show that basically variants arising in development are sufficient. You don't need selection, basically. You don't need strong selection, which was a kind of strong prior, but basically you don't need strong selection to get, that's actually inconsistent with these data on unrelated individuals, but variants arising in development under weak selection, weak or neutral, they're almost the same, are sufficient to explain the data on all three types of individual, unrelated, but crucially the dizygotic and the monozygotic twins. And so we think there was a lot of people spent a lot of time looking for sources of positive selection in clonal hematopoiesis. And we really think that this is not the right place to look. And a lot of people did this by DNA sequencing, but the point is, when you're looking at birth, you just haven't mutated enough to get a good clonal readout. So that you can do is you can be Sanger and you can spend like tens of millions sequencing things, but there haven't been enough mutations. But there's been enough flip-flopping of the methylation, because that is happening across the genome and it's happening early. And so we show that variants arising during development under weak selection are consistent with the lifetime clonal dynamics of HSCs. And yeah, the clones under weak selection, they can tend towards fixation given enough time, right? This is just PopGen, and a human lifespan gives you that amount of time. So I'm happy, I didn't do justice to that. I'm happy to, of course, go into more details as is your want, but I just firstly must not miss thanking all the people who really performed all this work in my lab and collaborators at USC and in Germany at the Fritz Lippmann Institute, which is the lab conducting the multiomic experiments in hematopoietic stem cells. And thank you for your attention. I'm happy to take more questions.
The situation where dominant
Yeah.
called CHIP.
Yeah.
There are mutations associated with CHIP.
Yes.
So are you saying those associations are sort of minor component, or unnecessary to explain what you see, or where do they fit?
So CHIP is clonal hematopoiesis of indeterminate potential, and yes, and it is often driven by mutations in specific genes that we see, DNMT3A or TET2. But there was a lot of work that tried to find driver mutations, like for CHIP or preceding CHIP. And there was like a lot of literature said that like these like that there's missing drivers. Okay. So even where yes, you have these like commonly mutated genes in CHIP, but like by doing like the phylogenetics of the HSCs, you can ask how much what's the strength of selection, and there were not enough kind of drivers, be it DNMT3A or other genes, to explain the clonal like composition of the bone marrow niche in old age. So our question:
you're saying it's going to arise without these mutations by weak selection,
yeah,
but you don't fit the mutations that you're on.
That's right. So we're like agnostic of the genetic state, but we're just saying that like you don't need to look for, like a lot of people were looking for the source of positive selection, and we're saying that like given these crucial factors of like having variants arise before birth, it's specific to the dynamics of the hematopoietic stem cell niche, which you have this population of cells, a few tens of thousands, roughly constant throughout life, but obviously experiencing a very large growth phase during development with the formation of hematopoiesis. So if you have a variance arising then even under neutral or a weak selection model, over the many decades of a human lifespan, that will give you enough time to reach the clonal compositions that you would expect in the data.
I want to clarify that parallel clonal composition of twins.
Does that imply that there's some variable shared event opening of that is very initiates first shared bonds if I sprinkle the data? So the data don't have like yeah, so either and like so epigenetic shared component or like a shared clonality, so basically the clones which are evolving together.
And those clones were there early on and just took a while to.
I mean, that's
Show that
that's the big
pattern 9 years
question,
later.
right? Could some other mechanisms, could those clones arise in the life of those twins after birth, after independence, through some other genetic mechanisms, or were they there from the start? Like, we can't detect that. But we just see, and of course there's a huge there's monozygotic twins which have relatively uncorrelated clone compositions also. Like, the variance is larger than for unrelated individuals. But it's like a subset of monozygotic twins that have some shared component. And we think the most consistent explanation is that genetic variants that are really before birth.
And they were supply somehow.
Yeah. Or they just, I mean, they could have very, very weak selection. It could be neutral or very weak. So they just take a really long time before you can see.
Right, if they were neutral, share stochasticity between twin pairs, they would correlate the time of the total history.
Yeah. So we think that they have their non-neutral, but just
Yeah,
very,
they have some
Deterministic.
yeah. But the crucial thing is like that because if you have this shared blood, if you're monopolyonic twins, so in the growth phase, like before birth, these variants can already grow up to some fraction of the blood, and then they could be the blood is very polyclonal at birth, so they're not going to be a large fraction, but there might be some ratio that's already like maintainable from birth. Thank you. Yes.
Okay, I have one question and then one question.
I just want to clarify, you talked about the last regression model with the gene specific. Can you clarify what you mean by the gene specific?
Yeah, so that's the gene specific parameter, like lambda is like the characterization parameter, and the regression, and basically we're varying that lambda based on the interactions that we're learning. And the reason for doing that is because if you have a fixed first parameter, then you will have some transcription factors that have no targets, and others that have many.
Nice. Okay.
And so by varying that, you balance you basically do slightly worse in terms of the error than you would otherwise, but you have more non-zero correlations selectively for certain gene interactions. That's the idea.
My bigger picture question is, how do you get confidence with a larger frame, like eating?
Yeah. I presume here you mean like more like biological conceptually scale rather than technically. And yeah, I don't think that would necessarily be like the framework the pop infer framework I would use that for classification, because you need cell state transitions that happen over the time frame of cell divisions, and you can think about one or two or a few cell divisions, but I don't think you can think about the stem cell dynamics over years on that same thing. So I think that I do think that about that a lot, like coupling with timescales of stem cell differentiation and aging. So I'm happy to chat more about that, but I don't think that this does not apply. And the timescale we have many cell divisions. It needs to be focused.
Thank you.
It was really nice meeting
Yeah, I like that. Sure. Yeah. Right. Yeah. Good luck with your paper. Yeah.
Credit.